Hello friends, welcome to Sam Biology class. Today we shall discuss about mismatch repair. Before that, I request all of my friends, please subscribe my YouTube channel and watch, like and share my videos with your friends. Mismatch repair MMR is a process used to correct any replicational mispairing that skip proofreading by DNA polymerases. The MMR system, discovered by Paul Mudrick, can also correct insertions or deletions of up to 4 nucleotides that arise from the slippage of one strand relative to the other in the active site of the DNA polymerase. The importance of mismatch repair is indicated by the fact that defects in the human mismatch repair system results in high incidence of cancer, notably hereditary non-polyposis, colorectal cancer syndrome, colorectal cancer syndrome that affect several organs and may be the most common inherited predisposition to cancer. Mismatch repair in E. coli is carried out by 3 proteins such as MUTS, MUTL and MUTH. This is a DNA molecule which is hemimethylated. Hemimethylated means only one strand, the parental strand, is methylated and the daughter strand is not. The parental strand has the correct base, but the daughter strand has incorrect base. This gives rise to a mismatch. First of all, a homodimer of MUTS binds to a mismatched base pair or to unpaired bases and then binds a homodimer of MUTL. This results in the formation of MUTS2 MUTL2 complex. This is the complex which consists of a homodimer of MUTS and MUTL. In the next step, in a ATP-dependent process, this complex translocates along the DNA in opposite direction, thereby forming a mismatch containing loop that is closed by the MUTS2 MUTL2 complex. Energy for this translocation along the DNA comes from the hydrolysis of ATP. In order to correct replicational errors, the mismatch repair system must distinguish parental DNA which has the correct base from daughter DNA which has the incorrect although normal base. Mismatch repair system in E. coli distinguish two DNA strands because the daughter DNA remains hemimethylated until methyl transferases until methyl transferases such as dimethyltransferase have had enough time to methylate the daughter strands. After that, on encountering a hemimethylated GATC palindrome, the MUTS2-MUTL2 complex recruit MUTH and activates this endonuclease. Activated MUTH introduces a single-stranded nick on the 5' side of unmethylated GATC sequence. This is the unmethylated GATC sequence, and this is the 5' side, and that is the 3' side. MUTH, after its activation, introduces a single-stranded nick on the 5' side of the unmethylated GATC sequence. This is the position of cleavage. Only the daughter strand which is not methylated is cleaved. UVRD helicase, which is also involved in nucleotide excision repair, separate the parental strand from the daughter strand. Then an exonuclease remove the defective section of the daughter strands. The removal of the defective section of the daughter strand by the action of exonuclease results in a gap which is then filled by the action of DNA polymerase III holoenzyme. This nick is then sealed by the action of DNA ligase. In eukaryotic cells, there are several homologues of MUTS and MUTL, but not of MUTH, Eukaryotic cells must therefore adopt some other mean to identify the daughter strand. One possibility is that the newly replicated DNA strand is identified by the presence of yet unsealed nicks between Okazaki fragments. DNA resynthesis is probably mediated by DNA polymerase delta. So friends, in this way, different types of replicational errors are corrected by the mismatch repair system. For more updates, subscribe my YouTube channel Sam Barus Plus.